This is Mahmoud Ali Musa doing biotech assignment with my colleagues Ryan, Jiyoung, and Jeeves. I will do the introduction. Our topic today is about tissue culture. Tissue culture is basically placing a specific cell or tissue in a specific medium which is optimized for optimum growth and survivability. Tissue culture firstly appeared in the beginnings of the 18th century. However, the foundation was placed by its godfather in 1902 by Gottlieb Hadderpelt. Next. Tissue culture can be seen in every field in medical and industrial world. Tissue culture is extremely needed to produce large amount of specific products. For example, you place a specific dead virus or bacteria in a plant, then you tissue culture to gain a large amount of it, and it can be used to produce specific medicine like antibodies and several other medicines. Also, one crucial use for tissue culture, it is very important as stepping stone for any research or medical examination, because you need to increase the size of the and the amount of the specific targeted microorganism, however it is harmful or useful. For example, you can test and do testing on a large amount of COVID-19 cells that was tissue culture to be able to produce a vaccine. Now we go to Jeeves.
Thank you, Mahmoud. Now I would like to go towards the advantages of tissue culture. There are many advantages of tissue culture. One of them are a large number of clones can be produced from a single parent plant. Secondly, it enables us to select and culture the desirable characteristic traits. For example, a mango that is fruity and another mango which is big and ripe. We can combine it and make itself take a stem out of it and produce tissue culture based on top of it. It also has short production time with long generation time. Fourthly, reproduction of plants is possible even with the absence of seeds or necessary external pollinators to produce seeds. Other than that, it frees us from the restriction of growing plants only in certain seasons. This also include countries such as United States of America, towards the western side, I mean. Next. Other than that, endangered plant species on the verge of extinction can be saved with the tissue culture procedure, such as plants from Malaysia which are Rafflesia, and other plants which are in western side which cannot be saved due to the seasonal and deforestation, which causes many plants to wilt and die. This is a major crucial effect on the history of mankind. Other than that, only a small amount of initial plant tissue is required. This doesn't only include the stem and the roots. This also includes the leaves, the xylem tissue and the phloem tissue. Other than that, the new plantlets and plants are more likely to be free of viruses and have resistance against diseases such as many diseases. The process is not dependent on the seasons and can be done throughout the year, but be aware that it should be under science labs for a certain period of time before it can be let out in a field. Finally, you need only a relatively small space to perform this process. I would suggest 10 times the plant in 1/10 of the space. Thank you. Now I'll pass my slide to Ganzi Yong.
Good morning to everyone. So now I'm going to present on how tissue culture works and how it benefits the society. Firstly, tissue culture has been refined to suit the needs of orchid species and hybrids known to grow well. In Malaysia, the ornamental and cut flower trade is a substantial source of foreign exchange and additional income for small growers. Other than that, in Thailand, tissue culture is used to reproduce slow-growing and environment-sensitive orchids. Thailand is the leader in tissue culture in Southeast Asia, producing 50 million plantlets a year. Most of these are orchids, which have helped the country become the biggest exporter of whole and cut orchids in the world. Next. Lastly, micropropagation by shoot culture technique has been developed for the mass propagation of banana. In the Philippines, this is used as a control approach to viral disease in banana, such as banana bunchy top virus (BBTV) and banana bragg mosaic virus (BBRMV), which are commonly spread through propagative material. Now I will pass the slide to Ryan.
In conclusion, tissue culture is therefore of great significance in biological studies due to a wide range of applications. The processes involved in tissue culture may be complex, requiring a lot of care to avoid such effects as contamination. Because of the complexities that might be involved in some of the steps, this may not be an experiment for everyone. Thank you.